Hello students, today we are going to start with the new topic which is chromosomal theory of inheritance, which is from your chapter 5 with principles of inheritance and variation. So let us start with what is actually chromosomal theory of inheritance and what does it says. Okay, so shall we start now? See, this chromosomal theory of inheritance, this is the practice done by three scientists who has done re-experiment on the Mendels laws. They were Hugo de Vares, Correns and von Tesmark. They had done independently this experiment and have tried to prove the theory which they have performed. So by this time, when they were doing their experiment, by this time microscopes were advanced and well developed. So at this particular time they have got to know that there are some chromosomes which are present in the human body, and these chromosomes have gene on their body. Like if this is a chromosome, this chromosome contains gene on its body. This is a gene 1 and this one is gene 2 and this one is a chromosome. So they have proved that there are not only the alleles which transfers the characters from one generation to another, but they have proved this also, that there is a chromosome which contains gene on its body surface and then the characters are transferred to the next generation. So they have proved about chromosome, and they have proved this also, that the structure in the nucleus which is appearing, it is having double number of chromosomes in itself, and then it further divides also at the time of cell division. Means these chromosomes are present in a pair, and when the time of a cell division come, they again divide themselves, they do reduction division. So this was given by the three scientists who are called as Hugo de Vares, Correns and Tesmark. Further, in 1902, they have done this experiment in 1900. But further, in 1902, there were the two scientists called Walter and Boveri. Their full name is Walter Sutton and Thordy Boveri. So together they are called as Sutton and Boveri. So they have done their experiment and they have noted down, they said that the chromosomes shows movement during the meiosis. Chromosome shows movement, that means chromosomes divide themselves, and after division further at the time of a gamete, when the gametes fuse at the time of a zygote formation, further they again join themselves with the other chromosomes of their same pair. So this was given by them, by Walter and Boveri in 1902. On the basis of this experiment, on their activity, they have given a theory called chromosomal theory of inheritance. And according to this law, according to the theory of chromosomal, according to this theory, they said few things. Amongst them, first one, what they have said was all the hereditary characters must be with the sperm cell and the egg cell. So they said that whatever characters are there, means the features which we are having, like the eye color, hair color, skin color, these all should be with sperm plus egg cell. Why? Because these are the gametes which are going to be transferred to the next generation. Okay. So this was the first theory which they have given, and in this it explains. Secondly, they said the hereditary factors must be carried by the nuclear matter. So they said heredity factors must be carried by nuclear material. So they said that it has to be transferred, and it has to be with the nuclear. If it will not be present with the nuclear or nucleus, then how come they will go in the chromosome? And if not on the chromosome, how the gene will work? So it is must that it should be on the nucleus. So because in nucleus there is a chromosome, and in chromosome there is on chromosome there is a gene which is actually giving the characters. Third point, they have explained that chromosomes are also found in pairs like the Mendelian alleles. So they said that the chromosomes are in pair. So the chromosomes, we have seen that they are always occurring in a pair. When in body it is there, only in the gamete they are single. Otherwise they are always 1 and 2 in a pair. So this they have proved. Fourth point, what they have explained, that was two alleles of a gene pair are located on homologous sites on the homologous chromosome. This I have been explaining you several times, that on the homologous chromosome, chromosomes are going to be located on homologous sites. Okay, so what does it mean? It actually means, suppose this is a chromosome and its another pair is this chromosome. So for an example, at this point which is showing locus, which is 401. So at this location, if there is a gene which expresses, this is a gene, okay, if this expresses eye color, so the same way on this chromosome, which is, because they both are the homologous, right? So this chromosome at same position, it won't be going to be on 5.1 or just 4.0 or just 4 point. It is definitely going to be on the 401 place, and here it is going to explain about the eye color. So this is about the homologous chromosome and the where homologous genes are going to be kept at the same place. Next point which they have explained was that the sperm and egg have haploid sets of the chromosomes. So sperm plus egg have haploid chromosome. This means that it is not at all going to be in a pair. This is just going to be simple singular in number. Why simple singular? Because already segregation process has been taken place over there. When the gamete was getting formed, at that time meiosis happens, and because of the meiosis 1 and 2, this has got segregated into just one single part. So this is now just one single chromosome present over here. So it is must that these two are going to be haploid chromosome. Next point, what they have explained, they said that genes are carried on the chromosomes. Genes are on chromosome. So whichever chromosome is there, whatever number of a gene, whatever type of a gene it has been allotted, it is going to carry that. Why it is going to carry that? Because when sperm will take this to the egg cell, there they are definitely going to be carried out. So if chromosome is carried out, of course the gene is also going to be carried out. Only the matter will be there that which one is going to be dominant and which one is going to be recessive, right? Next, which point he has explained was that the homologous chromosome synapse during meiosis. So synapse during meiosis, what? Chromosomes which are homologous. So he said that homologous chromosomes are going to synapse themselves and get separated to pass different cells. This was his explanation. He said that first these chromosomes are going to find out their homology and they will pair with it. Further, after getting paired in the G2 phase, further in the meiosis 1 they are going to be attached at a polar region, and further in meiosis 2 again they will get separated from their pairing, and it will from 1 it is going to become 2, and from 2 further in meiosis 2 it will divide, and from 2 it will become the 4. For example, if here one cell is there, this is going to divide itself into 2. Why this nucleus is getting divided? Because it is having chromosome. This will further divide into 2, and this is further going to divide into 2, and they all are having this chromosome in it. Okay, so here it was 2n, but here it has become n, and already here it is going to be n only. Okay, so this was his explanation. Now let us see this with one more example. You might have seen in your NCERT textbook that there is a figure. I'll try to show you this figure with this. Hope that this figure is quite very much visible to you. So here in this figure you can see in a very nice way that here at this place, this is a actually example which is explaining you about the segregation and independent assortment which happens because of the meiosis and the germ cells are formed. What is germ cell? So germ cells, these germ cells are formed over here. These germ cells are actually going to make further egg cell and sperm cells at the respective sites wherever they are present. So they are going to be responsible for the N, means for the haploid gametes. So let us see here at the G1 phase and G2 phase what happens actually. So here the chromosome which was there, it finds out its homology. So here they have found their homologous pairs, right? So which one was the homology? Now here at this place they are getting joined, they are going to get attached with each other, and here in the meiosis ana phase 1 now they are going to get assembled at one polar area, and now at this place division will starts happening because this chromosome is going to get bifurcate from its centromere independently. The word independently explains that this particular part of a chromosome is not going to have any problem or any issues with this part. Whatever characters this strand of gene is having, it will take it and it will get separated from here, and this part of the chromosome will again take its own part and get separated out from here. So the same way they both are going to get separated by themselves. So at this place by this time they are the 2n. Now here it is going to get converted into n. Now at this meiosis 2 phase you can see very nicely that here they have joined, but here at this place they had got separated. They are differentiated. and here this strand is also got separated. Now they are having the characteristic features whatever they have received. And here in the germ cell, which is completely N, you can see over here, this is also N and this is also N. So here from 2 to 4, from this to 1 they have become 2, and from this one they have again become 2. So from these two you are receiving total 4 number of the germ cells. So here it was in the pairing, but here they are the segregated one. So this is actually happening because of the cells division. This process happens in the meiosis, and that's why it is also called as a reduction division. Now further these germ cells will start producing their gametes, right? So this was the experiment done by the Walter Sutton and Theodore Bavry. They have given this their theory. Further, there was the Morgan, who did experiment on the chromosomal, this theory of inheritance which he has received. On this he has done the experiment called the organism Drosophila. Drosophila we also call as a fruit fly. So he did his experiment on the Drosophila, which is called as the fruit fly, or like that. So the experiment, because of his experiment, he has given some laws, some theories, and that is called as the linkage and recombination. Now let us understand first that what is this linkage and what is this recombination is saying. Actually, linkage word means to add or to connect. Fine. But the word combination means that two things have joined, right? Combine karna yani ki jodna. So it's saying that two things have combined, and word is used here re. So that means again. So something has joined again over here, that is called as recombination in our genetics, and to add or to connect anything is called as linkage in our genetics. So hope that these two words are now very much clear to you. Now let us see that what was and why Morgan has chosen this Drosophila for his experiment. See, the Morgan, he has done his experiment just like the dihybrid cross how Mendel has done. So the same way how Mendel did, he has performed it as a dihybrid cross, and this was done on the fruit fly, which is also called as a Drosophila, right? So in this experiment he has chosen the sex-linked chromosome, and this sex-linked chromosome is called your X chromosome, right? So on this X chromosome there were the certain genes which were present, and he has to study about this particular gene only. So Morgan has chosen fruit fly for his experiment because he can easily grow in his lab. So easily in his laboratory with the artificial medium he can grow them, he can develop them. The life cycle was just 2 weeks. So that was also fine, that life cycle is of just only 2 weeks, so he can get a lot of Drosophilas. Then third point was in single mating, he was able to get large number of organism. Means at just one single mating he used to get a lot of eggs which develops into the Drosophila. Next, there was a very good point in that the sexes were identified easily. Why the sexes were able to be get identified easily? Because the male which was there, the male was smaller and the female was bigger in size. So for that reason it was very much easy for him to find it out and to see that which one is the male and which one is the female. The next reason behind this selection was that they was able to show many heredity variations. Okay, so several heredity variations were able to be shown in them and was used microscope. So under the microscope a lot of characters actually they can see in these Drosophila. So for this reason he has chosen this. So now let us follow, let us see that what was the actually linkage and recombination and how he has proved that. So if you have to define, you can say about the linkage that they attributed this due to the physical association of two genes, and then the word term linkage was coined. This is actually the physical association of genes on a chromosome, and the term recombination is to describe the generation of non-parental gene recombination. So for this, now let us see one of the example and then understand. Before that, I will tell you one thing. See, for example, if this one is a chromosome, right? On this chromosome, here is one gene and here is the another gene. Gene number 1, gene number 2. These two genes are close to each other, right? So if they are very much close to each other, then it means that their addition or their linking is very strong. So if their linking is strong, then it means that we are not able to divide them easily. So the characters which are here at this place, close genes, will be always carried to the next generation, but there will be a problem if gene number 1, 2 is here and gene number 3 is somewhere here. So the distance, if you will see between these three, then that means this gene number 3 is quite far away. It's far from here, this place. So that means it can be easily broken out, or it can be easily not get transferred to the other condition or other offspring. So these two are very strong, very determined and very much capable to be get transferred, but this one is not, right? So now let us see this same thing with one of the example, with one of the diagram which is given in your NCERT book also. So just let's have a look of it. I think, okay. Now I hope that this is visible to you people. Okay, so now let us see. Here in this diagram, it is explaining there are the two different possibilities are shown. This is the cross A and this is the cross B. So two different cases are over here in this diagram. This is a parent's body. Okay. Now in this parent body, one of the parent is having yellow color body and white color body and yellow eye color. The other one is wild variety. Okay, so this is the parent's body. Now here in this you can see these two genes are very much closer to each other, right? But actually here in just one of it there is a gene located, and this one is a vacant where there is no gene present, right? The same way in this, when they two have gone through the cross, now let us see what is going to happen actually. The genes which are very much closer, it is not going to be broken down easily, right? So the chances of them going to be in the next generation is maximum. So now in F1 generation they have received that this one feature of wild type has been transferred in the chromosome of the one child, and here in this one of it has got transferred over here. So the chromosomes have interchanged themselves, right? So here this one has come, and at this place instead of this one has come, right? So because of this has happened because of the gametes, remember? So because of that this interchanging have got here. Now you might be having some confusion. If you're having some confusion, then you can remind yourself about the cross which we do in a Punnett square, right? So keep that also in your mind, and with that help you can solve this. Now in F2 generation we are reaching. Now in F2 generation what is happening over here? See, the maximum of them are having this kind of a gene present on their body. Here this one is also having, this one is also having, here and here. So what is happening? We are getting just one wild type and two are here which is white type. This is actually F1 generations. Gametes have got transferred, and here this one is yellow and this one is white, but maximum of them are having in their genotypic ratio about these chromosomes, right? And this one has got changed to all these places. Same way when this has followed for this F2 generation is further seen, then again here you can see that same gene is present everywhere, and only here at this place this is vacant. Only at this one is the wild one showing, rest all are showing different types. Means only one is wild, rest all are having the pairs, the characteristic of this yellow and white type. So here it is like 3:1 ratio, not like Mendel. What did he said in his dihybrid cross? In his dihybrid cross he said the result is going to be 9:3:3:1. But here we are getting maximum in the type of a yellow white, and just one is here which is the wild type. So it does not follows Mendel's rule, Mendel's explanation that the result will be always 9:3:3:1 when dihybrid cross is done. So here it cancels, here it rejects the following of Mendel's theory. Same way in this experiment also you can see the white miniature, means short-heighted Drosophila, was crossed. Wait, I'll show you. Is here it is crossed with the wild type, and further this is going to be the F1 generation. Now in F1 generation you can see the miniature one is here, white is here, and this one is the wild type, right? So if they have got the cross, this mini wild type is here, and it's one of the chromosome has shifted over here because we are making the gametes, right? So here it has got interchange over there. Now in this F2 generation what is happening, actually the parent's body is about 62%, and the recombinant type is going to be just 37 points, means something which has got differentiated, which has got the from the ancestors, that crossing over is again done, or the characters have been again changed over there. Otherwise the same characters from the parents which are there is got transferred in the offsprings, but here something different has found, something different has got changed over here. At this place you can never see at this much of white part is there, that means this one is going to be quite whiter than the parent's body, because nowhere this much long gene is present. कहीं पे भी यहाँ दिख रहा है इतना लंबा सा नहीं दिख रहा है ना. So this one is the only one. So this is going to show recombination much more. Same way here, this kind of gene is nowhere present in this body. So same thing is going to be here also, right? So that means the parental characters is coming about 62% and recombinant feature is coming 37 point. But if you will tally with this type of a cross, in this case, here you will be able to see that recombination type is just 1.3%, but the parental character is maximum over in this. So what it is actually explaining, it is explaining the linkage result of two dihybrid crosses which was done by the Morgan. Cross A shows the crossing between gene Y and W, and the cross B shows crossing between gene W and M. M means miniature. Here dominant wild type alleles are represented with plus sign in the subscript. So wherever the plus sign is mentioned, it is explaining you about the dominant feature. So now you must have gone through this, that what this is actually explaining. Just go through this once again, keep your NCERT book in front of you, and again you watch the video and try to read by yourself also. So your problem will be sorted out. Have a good day.